Hi, I'm Annah-Ololade Sangosanya, and I'm a biologist by training. I specialized in microbiology, synthetic biology, and fermentation processes during my master's at the National Institute of Applied Science in Toulouse. And after my master's, I decided to continue my studies at Fab Lab Barcelona, at Fabrica de Mi Barcelona. It's a postgraduate in textile and technology, during which I specialized into mycelium materials and digital fabrication. I am now an independent researcher focusing on mycelium materials and their applications in fashion and textile recycling, and I'm working in this mycelium materials lab that I developed with Jessica Diaz in the Paraguay Project Studio. Welcome to my lab. Our society is in desperate need for environmentally sustainable solutions to tackle issues that are related to ecological destruction and resource scarcity. And that's why there has been a clear rising trend for researchers, makers, and designers to turn to biology to broaden the sustainable material base. In today's masterclass, we will explore the dynamic relationship between fashion and new lab-grown materials, and how biology, biochemistry, genetic modification, and industrial processes converge to drive technological advancements in the fashion industry. I'm thus first going to give you a brief introduction to biology and its concepts so that everyone, even people from a non-scientific background, can understand those new technologies. And once the fundamental concepts have been explained, I will break down the different types of materials that can be considered lab-grown and explain how they are made. Don't worry, I will be giving examples all along the way for a better understanding. And finally, I will be explaining a bit deeper in what my work consists in, the challenges I can face as a maker, and how we can still innovate in that field. Let's get started. So before starting to dive into the subject of lab-grown textiles, the first important thing is to understand how they can be made. So for that, let's go through a short introduction of biology and some of its fundamental concepts. So the first thing you need to understand is, what is a cell? So a cell is the smallest unit of life. Everything that is alive is made of one or multiple cells, whether it's us humans, animals, plants, fungi, bacteria, everything. So there are many different types of cells. For instance, our brain cells are very different from our skin cells, but also our skin cells are very different from bacterial cells. The main difference between the different types of cells relies in the DNA. Every cell has its own DNA, which can be considered as a program code. The cell just executes what the DNA code tells it to do, which is why we can consider cells as microfactories executing the orders from the DNA. Cells from simple organisms like bacteria can grow alone, and cells from more complex organisms like animals, plants, or mushrooms can grow in complex structures, which are called tissues. This behavior is also dictated by DNA. So another powerful thing the DNA can do is tell the cell to produce certain types of molecules. So to sum up, depending on the DNA that it is given, the cell can do various things such as producing different kinds of molecules, grow alone, or grow in cell tissues. And if we change the DNA the right way, depending on the DNA that it is given, the cell will have different behaviors. So this is how genetic modifications work, and this is how they can help us produce different kinds of molecules or have organisms grow the way we want them to grow. Now that the concept of DNA and cell is clearer, we can really dive into the subject of lab-grown textiles. So there are two different types of lab-grown textiles, two main types of lab-grown textiles, the ones that are made of cell tissues and the ones that are made of the molecules that the cells produce. Lab-grown materials made of cell tissues are leather-like materials. Using a cell tissue is the same principle as traditional leather, as it is made of the skin cells from animals, mostly cows. Lab-grown leathers made of cell tissues are grown on a flat surface to replicate the principle of a cow hive. The cells used in this process can be animal skin cells grown in specific conditions, offering a cruelty-free alternative to classic animal leather. But leather-like material made from cell tissues can also be made from different types of cells, like fungal cells, which is the principle of mycelium leather. Fungal cells, when put in specific conditions, can grow just like animal tissue and make fungal skins that resemble leather in terms of visual, feels, and mechanical properties. However, growing cells is a complex process, especially for beginner makers, because it requires the adapted expertise and heavy equipment, especially when exploring larger scale production. The second types of lab-grown textiles are more accessible to makers, as they are made from the molecules produced by the cells. These molecules, in the case of fashion applications, are usually polymers. Cells can produce various kinds of biopolymers, including cellulose, collagen, silk, or alginates, and these polymers can be treated in two different ways by us humans, so we can cast them or spin them in threads. Casting those polymers allows for the making of materials resembling leathers and plastic, whereas the spinning of the polymers allows for the making of threads and yarns. So, casted polymers is a technique that is way more accessible to makers, as the process can easily be replicated in a kitchen, and in most cases, the raw materials are available commercially and inexpensive. In some cases, the cells producing the polymers can be grown in simple setups, or the polymer itself can be purchased from companies who extracted it from waste sources. So examples of casted lab-grown materials are Kombucha Leather, but also alginate, agar, or gelatin bioplastic, and collagen leather. In the case of kombucha leather, the bacteria and yeasts that are present in the drink can produce cellulose as a byproduct of their fermentation process. They feed from sugar and produce the polymer that accumulates on the surface and that is called SCOBY. The SCOBY can be harvested, dried, and post-processed to be used as a material for fashion applications, just as featured in the Regrow project. In the case of kombucha, the microorganisms present are naturally producing cellulose. But companies have also been exploring the possibility to genetically engineer some microorganisms to make them produce other polymers. This is how the company Modern Meadow made a bacteria producing collagen, which is the main component of leather, to be able to make a cruelty-free, vegan, leather-like material. But there are other ways for us makers to make lab-grown bioplastics and leather-like materials. Agar, alginate, and gelatin are commercially available polymers that can be used to make casted biomaterials. This process is simple and easy to implement, as it usually involves dissolving the polymer in water with glycerin, heating, casting, and drying. In this case, the role of glycerin is to be a plasticizer, so the purpose is for it to bring flexibility to the material. Various recipes can be explored with different ratios of glycerin to achieve different flexibilities, but we can also explore the addition of colors or fibers for various visuals, patterns, textures, and resistances. The use of bioplastics in fashion is being explored by various makers thanks to the versatility of the achievable textures, from latex-like to leather-like. But it is also biodegradable, and we can also achieve different types of colors, including color fastness. If you are interested in exploring the universe of bioplastics, I invite you to try some of the recipes described in the Fab Textiles Bioplastic Cookbook by Margaret Dune. It is important to note that all of these polymers can also be extracted, purified, and then spun into yarns using processes that already exist for man-made cellulosic fibers, for instance, viscose, tencel, or rayon. And so this gives lab-produced polymers a huge advantage because they're easily scalable for industrial applications. This way, bacterial cellulose has been spun into yarns by the company Nanolose, but algae yarns were also made by the companies algaing, and calcium. While these processes are a bit more complex to beginners in the field, I invite more experimented makers in the biomaterials field to try spinning their polymers following this video. When diving a bit deeper into the world of biology, we can discover that everything can be made. As a reference, some even crazier companies like AmSilk, Bold Threads, or Spiber has been developing genetic modifications on yeast and bacteria to make them produce an enhanced protein resembling spider silk with enhanced properties. The Craig Biolab laboratories in the US have even implanted the DNA coding for this enhanced spider silk protein to be produced by lines of transgenic silkworms. But this completely outpasses the maker's scope. As a maker with a background in biological engineering, my focus is the exploration of mycelium leather-like materials, and I have been investigating ways to produce various kinds thereof. So, for instance, the different colors and textures that different fungi strains can produce, and their mechanical properties. The main challenges I face in my everyday work is that this field is very new, and finding protocols on how to make those new materials can be a bit challenging, but this is the whole purpose of the research. However, bigger companies have been investigating ways to produce mycelium leathers and mycelium materials and have been filing a lot of patents to be able to exploit those technologies commercially. Those patents are available online and can be a source of inspiration when trying to get to know a new technique or developing new protocols. But they become a barrier as soon as us makers try to explore these technologies commercially. And the second challenge faced as a biomaterials researcher is that most of those materials are referred to as bioplastics or leather-like materials. Despite the environmental downsides that those materials feature, they are widely used for industrial applications and they constitute the standards in terms of mechanical properties, resistance to heat, and sterilizability. To date, no leather-like material can beat leather in terms of resistance, and calling them leather-like materials implies expectations of similar mechanical properties. One of the important axes to explore in the future is to shift the perspectives on biomaterials. Even if they are a powerful alternative for making environment-friendly products, they are not leather, they are not plastics, and they cannot be suitable for replacing those materials in every application. It is thus important to find new applications for those new materials and eventually replace the old applications with the new objects that we would be creating by gradually changing the vision on those materials, and that implies slowly changing mentalities. So by now you might be wondering how we as makers can have an impact on this complex field. Well, there are many ways of innovating at our own scale. First, we can try and explore new applications for those already developed materials by thinking out of the box and imagining different and new kinds of objects out of them. And even though I presented you some of the existing examples, there are many other biopolymers that can be explored for fashion applications. I told you about cellulose, agar, alginate, and gelatin, but we can also imagine exploring chitosan, casein, or keratin for fashion applications. Since this whole field is very new, including the values of distributed design into our practice will keep research going by allowing to implement innovation onto existing innovation and to be able to reach great results. So to sum up, in this masterclass, we learned the basic principles of biology applied to biomaterials and had an overview of the different existing lab-grown materials used in fashion. We also had a glimpse into the challenges those materials come with, along with the vast potential of innovation they offer. Now that we've done all the theory, I want you to get your hands dirty and try making your own sample of lab-grown material. So if you're more interested into bioplastics, I invite you to follow one of the recipes from the Fab Textiles Biomaterials cookbook here. And if you are more experimented with mycelium materials, I invite you to follow one of my tutorials to try and make your own lab-grown leather. Little samples like that. But if you're more a beginner, I'm now going to give you a workshop on how to make kombucha leather. So to make kombucha leather, you will need one box that you will thoroughly clean with alcohol. You also need 1 spoon. You will need tea bags. You will need boiling water. You will also need white vinegar, sugar, and non-pasteurized kombucha. It is very important that it's non-pasteurized because you need the microorganisms to be alive. First, I will be cleaning the box with alcohol. That's just for you to avoid having contamination in your box. Then you will add the sugar. So the sugar is 100 grams per 1 liter of water. So I'm making 2 liters, so I have 200 grams of sugar. Then you can add the white vinegar. So for the white vinegar, it's 100 milliliters per liter of water. So since I'm making 2 liters, I have 200 milliliters. Then I add 2 tea bags, and then I'll go get some boiling water. Now that I have my 2 liters of boiling water, I will add them to the bucket. So essentially, it's just like making tea and adding vinegar to it. So now I'm going to mix it so that the sugar gets diluted inside the water. And then I will set it aside until it cools down to room temperature. So once your tea has cooled down, you can inoculate it with some kombucha. So you really need to have non-pasteurized kombucha because if not, the bacteria and yeast will be dead. So you want them to be alive so that they can start growing inside the tea and start making the cellulose pellicula on the top. So basically, you can pour as much of the bottle as you want inside. I'm going to be pouring half of it. Careful for the gas. So once you inoculated your kombucha with the bacteria, you can close the box and let it aside to grow for a bunch of weeks. So usually you will start seeing results after 2 months. So this is a pretty long process, but trust the process, you will start seeing the pellicula on top after that time. And so this is going to be your kombucha leather. Then you can harvest it. So take it, you have to rinse it very well. And then once you rinsed it, you can dry it and apply some oil on it so that it stays flexible.